Once upon a time, there was a new girl at school.
My name is Jothika. Can I sit with you guys?
Well, that depends. Do you know anything about protein structure?
I love proteins. There are 4 different levels of protein structures, the simplest being primary structure, which is, of course, just the linear sequence of amino acids. Did you know that the linear sequence of amino acids contains all the information needed for peptide folding? And check out my primary structure fancy necklace. It's so fancy.
I'm so fancy. You already?
I love your fancy necklace. Of course you can sit with us.
Yay!
Hey, our favorite level is secondary structure.
And my name is Fiker, and I'm really interested in alpha helix. Alpha helix is fundamentally important secondary peptide structure, and it is a right-handed coil. It is very stable structure due to hydrogen bond, which is formed between the NH group, the peptide NH group, and the carbonyl group. One turn of alpha helix represents 3.6 amino acid residue, and the hydrogen bond is usually prior to the alpha helix, and the R group is outside, pointing outside the alpha helix.
And my favorite thing is beta sheets, and they happen every 2 residues, so they're a little more stiff than alpha helices. And sometimes they can be anti-parallel and have parallel hydrogen bonds with hydrophobic residues on only one side, and sometimes they can be parallel with non-parallel hydrogen bonds and hydrophobic residues on both sides. And if the beta sheet has prolines and glycines, sometimes they can make a fold every 4 residues into a beta turn.
And we love tertiary structure, which is a 3D structure of a polypeptide chain.
Hey, I'm Rebecca, and I'm really interested in globular proteins. There are these 3-dimensional structures that contain large amounts of alpha helices and beta sheets. Both of these make up the core of the protein, folding into a compact form and is stabilized by both polar and nonpolar groups. Hemoglobin is actually an example of a globular protein, and those are found in red blood cells.
Hi, I'm Danielle, and I love fibrous proteins. Fibrous proteins generally have structural roles in cells, are insoluble in water. Alpha keratin, fibroin, beta keratin, and collagen are all examples of fibrous proteins. Alpha keratin contains multiple alpha helices and are subsequently strong yet flexible. They can be found in both fingernails and hair. Beta keratin and fibroin utilize stacked antiparallel beta sheets as their secondary structure. Lastly, collagen utilizes tropocollagen as its basic unit, which is could pose 3 intertwined polypeptide chains and a right-handed helix. Lastly, these tropocollagens then arrange in long sequences with gaps in between, and these fibrils can be found in bone and cartilage.
My name is Jesus, and I like transmembrane proteins. My favorite thing about them is that they have hydrophilic residues on the inside and hydrophobic residues on the outside. So it's kind of like a rain jacket where the hydrophobic, water-resistant residues are on the outside. Also, one quick fact about proteins is that they are denatured by heat, pH alterations, chaotropic agents, and detergents.
Hi, I'm Justin, and I love quaternary structure. Did you know that they're like the highest level of protein structure that can happen? And it's because they go through non-covalent bonds that results in a gain of function and a stable and favorable structure that there's a lowered surface area to volume ratio. Well, they can associate in different ways. So this way, it's like the isologous association, and what it is identical binding sites will attach to each other, or there's the heterologous association where they basically just stack on top of each other. And that's why I just really love the quaternary structure.
1, 2, 3, proteins! Just the linear acid of linear acid. Recording.
Hi, my name is Justin, and I just love shit.
I got it from my sugar-rich daddy.